In this lecture, we're going to take a look at the kingdom fungi and some of the characteristics of the different groups of funguses out there. So, mycology is the study of fungi, and fungi are heterotrophic organisms. So heterotrophic means that they get their nutrients and their nutrition by consuming the nutrients that have been made by other organisms. And there are a couple of different ways that they do this. They are saprophytic, which means that they digest dead organic material and waste products, waste stuff. And they do this by secreting enzymes on that material and then absorbing the digested nutrients. The digested nutrients into their bodies. Some of them are parasitic, and they get nutrients from the tissues of living organisms if they're parasitic like this. But frequently they kill cells first before they can then digest nutrients out of them. So even when they're parasitic, many of them are still saprophytic as well. So heterotrophic versus being autotrophic, that's anything that photosynthesizes or chemosynthesizes. Those guys would be autotrophic. Most of the fungi out there in the world are multicellular, with the big exception being the yeasts. And yeasts are found as single-celled individuals. They have a cell wall. Usually it's composed of chitin. That's a polysaccharide with some nitrogen involved in it as well. But there are a couple that also have, that instead of having chitin as their cell wall, they have cellulose cell wall. And most of them synthesize and store up glucose in the form of glycogen. So they have glycogen granules inside of the cell. And then last of all, the yeasts, they have plasmids. Remember, those are those circular bits of DNA. And because they accept plasmids as well, then we can build plasmids that have the genes to build products that we want. And so we can turn these yeasts into factories that will produce specific organic molecules by genetically engineering some plasmids and then inserting them into the yeasts. And then as the yeasts grow and divide, the new offspring yeasts that bud from them or are the products of cell division will have those plasmids in them as well. And so here we can see a close-up using an electron micrograph of a bread mold. It's called Rhizopus nigrans. We'll see some more of this same species. This is an awesome set of images from a fellow who runs the microscopy program at a community college. I forget, it was like in Illinois or Indiana, somewhere. And he has these just fantastic pictures. So if you are interested in what microscopists can do and how they can look close up at a lot of cool stuff, check out this website because it's really amazing. Okay, so when we think about fungal structure, we first have to think about what the fungal body is. And so the term for a fungus body is a thallus. And for most funguses, the thallus consists of what is described as mycelium, a loose mass of these thread-like hyphal or thread-like multicellular structures that reach out and cover over dead or decaying material and can absorb nutrients from this decaying material. And those threads are referred to as hyphae. So they're just like these strings that are built of cell after cell after cell of fungus cell. So it's the hyphae that then release digestive enzymes onto the substrate that they're growing across and then can digest it and absorb the nutrients from it. And so what we're looking at here is the mycelium of a fungus. Here's how it was originally placed onto the agar plate. And then you can see these threads of hyphae that are growing away from that, the original blob of fungus that was placed onto the agar. So the the enzymes can also digest into damaged cells, and this lets parasitic fungi invade their host. So they can digest in damaged cells, digest into them, and invade the host as well. So the hyphae can be multinucleate, but what we see in these multinucleate hyphae is that there are these septa, these little cross walls that have pores that allow the nuclei and the cytoplasmic materials to move from cell to cell. So here, oh shoot, I didn't mean to make that red mark. So in this image here that I'm pointing to, there are a bunch of little red arrows. They're pointing out those septa, these little cross walls that can allow the fungus to, allow communication among the cells that are making up the hyphae. And they allow the nuclei to move along those cells as well. And in some, though, it's kind of cool. If one of the hyphae cell is damaged or it gets old and begins to malfunction, then there's this specialized organelle called the Woronin body, and that will block block the pore into the damaged cell, and then that prevents the exchange of cytoplasm and nuclei along that hyphal thread. So here's a generalized fungus life cycle. I'm not going to expect you to know the life cycle of the individual fungi group. I think that's a little bit beyond what this class is trying to get to. But I do want you to understand the sort of general fungal life cycle. So the bulk of what we see, of the mycelium, these thread-like hyphal material, is haploid plant cell, or a fungal cell, excuse me. So it's haploid. It has only one copy of each chromosome. That's what this N without a number in front of it is telling us. It's a 1N cell, has only one copy of the DNA in it. And they will go from spore, grows up into a adult that then produces spores, and that is the asexual life cycle going round and round. But it is also possible for the hyphae to bump into a hypha of the opposite sex, quote unquote. Usually we think of it as plus minus. And when that happens, they can grow together. So you can get these gametes that will grow together, two haploid gametes, they grow together, but the nuclei don't fuse. So the cells fuse, but the nuclei don't fuse. And that dikaryotic cell, that's called, so dikaryote means that there are two nuclei within the cell, will grow and divide, producing a whole colony of these dikaryotic cells. And then later on, their nuclei will fuse in a process that's referred to as karyogamy. So we have plasmogamy, that's when the contents of the cell, the cytoplasm, fuses. Karyogamy is fusion of the nuclei. And at that point, you have a diploid cell. So that's what we're seeing here is the diploid cell as a result of karyogamy of the two nuclei. So the gametes, they fuse via plasmogamy, but it's a dinucleate cell or a dikaryotic cell. It will grow and divide, producing a whole bunch of these dikaryotic or individuals. The two dikaryotic nuclei will fuse through karyogamy, produce a 2N diploid cell, and then that 2N diploid cell will undergo meiosis, producing spores. And those spores then will germinate, and then once again we enter the asexual cycle. So spores can result from either asexual or sexual reproduction. So with sexual reproduction, we get some new mixing up of all the different DNA that's in the population. With asexual reproduction, we're just producing exact copies of the individual. If it's an aquatic fungus, then it'll produce these motile flagellated cells. If it's terrestrial fungi, though, because they live in the terrestrial environment, which can get really dry, then they produce spores that have very thick protective cell walls around them to prevent them from drying out and dying. So when the spores germinate, they produce a single cell, and that cell divides or produces that grows and divides, or they may produce this germ tube, which will penetrate the spore container, the wall of the spore, and develop into the thread-like hyphae. So it could be that it produces these individual cells like a yeast, or it may produce a fungal mycelium with a bunch of hyphae. Okay, so why are fungi important? They're important because they do this job of decomposing. They break down and recycle dead material. We would be up to our eyeballs in, or beyond in, undecomposed wood and other plant material and animal bodies and stuff if the fungi weren't doing this work for us. Some of them are facultative parasites. That means that they can, while they may be free living, they are also capable of living parasitic lifestyle as well, killing the cells and digesting them of a living organism. And almost all life forms can be parasitized by fungi. But what we see is that fungi are not, there are no obligate parasites among the fungi. They can always make it living free and on their own. And then last of all, many of the soil-dwelling fungi produce microbial toxins. So they have the capacity to engage in antibiosis. And some of those toxins have been engaged or harnessed as antibiotics that we now use for fighting infections. So, lots of ways that fungi are important to humans and to their environment. So what does it take for a fungus to be able to parasitize and invade a host organism? Well, first it has to be near the host. So the spores, they are meant to be mobile. They can be carried by wind or by water or even on the surface of other organisms. So insects can carry fungal spores around, and other animals will be serving as a vector for that spore. Second, they have to have so first they got to get near the host, and spores allow them to do that. Second, they have to have the ability to penetrate the host. And so if it's a fungus that wants to parasitize a plant, the hyphae will form these little pegs, little projections that, as the fungus grows, will push through the plant's cell walls. Man, I've got all sorts of hiccups and burps and stuff going on here. Sorry about that. I apologize to all of you for my disgusting digestive system. So so they can push into and through the plant cell wall to allow them to invade the plant cell. And if it's going after an animal host, then they contain lysosomes, the fungal cells do, and those lysosomes can be released, the contents can be released onto the host to digest their way into animal cells. So they have the ability to digest and absorb the host nutrients. That's the third capacity. If they can't digest it, then they won't be able to parasitize it. And so as the cells that they are parasitizing die, then the fungi will invade adjacent cells and continue to work their way through the organism. Here's an example of that. This is ringworm, not really a worm. It's a fungal infection of trichophyton. And you can see why it's called ringworm. It grows in these sort of ever-expanding round kind of scarry-looking things. So a number of different fungal parasitic diseases. If it's plants, they can cause wilts and mildews. They can cause blights and rusts and smuts. So those are all the different kinds of plant fungal diseases. Smuts usually have lots of spores on the surface of them. Blights, like the potato blight, can take out whole crops. Rusts grow on leaves or on fruit. Mildews will definitely grow on fruit and on leaves. So lots of different plant fungal parasites. In animals, we see that they are a significant loss for agriculture. So growing birds like chickens and cattle and other even plants like grapes and other fruits especially. So in humans, though, we refer to fungal diseases as mycoses, and there are we describe these mycoses based on what it is that they are invading. So a superficial mycosis would affect, say, the skin, the hair, the nails, while a subcutaneous mycosis is going to go into the connective tissue, and could even from there, because there's a lot of fluid in connective tissue that drains into the lymphatic system, it could even spread into the lymphatic system. And then if it's a systemic mycosis, then that is one that is invaded internal organs. So it's made it into the lymph or into blood, and now is invading the internal organs of the body. Last of all, we think about opportunistic mycoses. Those are funguses that generally don't cause disease, but if for some reason a patient is immune depressed, either because they're artificially depressing the immune system because they're a transplant patient, or if they have some sort of immunodeficiency disease like HIV that has progressed to full-blown AIDS or some other immunodeficiency, then the fungus has the potential, since we can't fight it off, to become pathogenic or even deadly. So that, by the way, is called an opportunistic mycosis, when it normally isn't, but it takes advantage of the opportunity if the immune system is depressed. Okay, so how do we classify fungi? They're generally classified based on how they do their reproduction, whether or not there's a sexual stage to reproduction, and then once we can see that there's a sexual stage, how sexual reproduction is accomplished. And so classification with these guys is super complicated, because in some, no sexual cycle has been observed, whereas in others, because of something we'll talk about in a minute, dimorphism, it's difficult to match the sexual stage to the asexual stage. So, for instance, here we have Candida albicans. It's one of these organisms that shows this kind of dimorphism, where it has different shapes and structures when it's in different environments or taking advantage of different nutrients. So, for instance, on mucous membranes, Candida albicans is harmless when it is in this yeast-like morph of its body. So as individual cells that are budding and growing and budding, it doesn't cause any disease. But if it switches to the mold-like form, where it begins to grow fungal hyphae and begins invading into mucous membranes, it can cause diseases like thrush, which is when the fungus is in the tongue and the mucous membranes of the mouth, or vaginitis, with the infection of the vaginal mucosa. So frequently, these stages are studied independently by different researchers, that these morphs are, and so they even go by different names. So we just don't recognize that it's actually the same species. For instance, the fungus that caused athlete's foot, we call it, and also is responsible for ringworm and jock itch. It's called trichophyton when it's asexual, and arthroderma if it's sexual. So we get different names, we got different shapes. It's just hard to get it all organized. So there's still a lot of work being done, but with the advent of molecular biology, we are now able to look at the DNA and recognize when species are the same, despite their dimorphism. Okay, so there are three groups of fungi based on their sexual reproduction, and then we'll see that there's a fourth group that doesn't reproduce sexually. So the first group is the zygomycota. The zygomycota are the bread molds, so you probably have seen this stuff. Sometimes it's also called the conjugation fungi. So they have this complex mycelium of hyphae that all have chitinous cell walls. And here we can see the hyphae growing here. These threads are the hyphae. When we look at Rhizopus, that black bread mold, here you can see the reason why it's called black bread mold is because it has these fruiting bodies that are black. But the hyphae grow really rapidly, and they produce super lightweight, easily to transported spores. And those spores germinate, and they produce more of the fungal hyphae. But they can switch to sexual reproduction if one of these plus and minus strains finds each other, they find each other by chemical smell. I say smell, it's not like they have a nose, of course, but they notice chemicals and they grow toward each other, and they form that multinucleate cell, the dikaryotic cell, and eventually the nuclei fuse, and that produces a diploid zygote that can be enclosed in a really thick-walled, resistant zygospore structure. When the zygospore germinates and grows, then it'll form the sporangium that produces the haploid spores. And so here are the sporangia that are filled with haploid spores, and they will then germinate and grow more hyphae and eventually find their opposite plus to minus or minus to plus, depending on which they are, and can begin the sexual reproduction cycle again. So that's the zygomycota. The second group are the ascomycota, sometimes referred to as the sac fungi. There's something like 30,000 of these species that have been described, and they include what we think of as yeasts. Though it turns out, anything that we talk about as a yeast frequently doesn't have a known sexual phase. So they produce a structure called an ascus, and this is the ascus here. It's the result of sexual reproduction that undergoes meiosis and produces a bunch of ascospores. Inside of each ascus, there are 8 ascospores. As the ascospores germinate, then they grow hyphae, and when a plus hypha finds a minus hypha, they fuse, they form that dikaryotic hyphal cell, and that will then develop into a structure as those cells grow and divide, develop into a structure referred to as an ascocarp. So this is a little body that is filled with fungal hyphal cells that when that dikaryotic cell, the nuclei fuse, they form a diploid cell that then grows into the ascus that produces the 8 ascospores again. So here we see the ascospores germinating, producing hyphae that find each other. The dikaryotic cells grow and divide, forming the ascocarp, which then leads to the production of more of these asci that produce spores. So what are some important sac fungi? Well, there's a bunch that are important to food production. For instance, Penicillium notatum, that produces, while it's not a food, it's important to medicine. That produces one of the first of the antibiotics that we developed, called penicillin, of course. But Penicillium, the same genus, has 2 other species associated with it, Penicillium roquefortii and Penicillium camembertii. These are both involved in cheese making. Of course, Roquefort cheese is made with Penicillium roquefortii, and Camembert is made with Penicillium camembertii. Saccharomyces, and there are multiple species of this. That is the yeast that leavens bread and is involved in brewing alcohol and beer and wine. And then other ways that the sac fungi are important is because they produce disease. And so some of the mycoses they produce include that candida. When it's in the mouth, it's thrush. If it's vaginal, then it's candidiasis or a yeast infection. Trichophyton, which is the same, that genus produces athlete's foot, jock itch, and ringworm. Aspergillus, blastomyces, and histoplasma, these are all sac fungi that are opportunistic and can cause respiratory infections. So if the immune system is depressed, then they can set up shop in the lungs and create infections. So here we see bread with aspergillus growing in it, one of the bread molds, and they're producing conidia, which are the spore-producing components of the mycelium. And then last of all are the things that we normally think of as mushrooms. These are the club fungi, or the Basidiomycota. And they include mushrooms and toadstools, as well as rusts and smuts. These are generally fungi that attack grains and plant material. The life cycle of a basidiomycote begins with basidiospores that germinate. So we have spores that germinate, and they form a mycelium of hyphae, and that mycelium will grow through wood and dirt and soil and all sorts of stuff. When the mycelial cells find each other, and they are opposite, plus minus, then they will form these dikaryotic cells. The dikaryotic cells will grow and divide, and they form a different kind of mycelium. And if it's a mushroom-forming species, then this is where we see the basidium develop, the what we think of as the mushroom. And so that's the fruiting body, and eventually specific cells will, on that growing mushroom, usually around the gills, on the underside of the mushroom cap, the nuclei will fuse and produce basidiospores that will then germinate and grow into a fungal hyphae mycelium that's in the soil once more. So these are important agricultural products. We love to eat some mushrooms. And then some of them also produce some pretty deadly chemicals. So Amanita is the angel of death mushroom. Claviceps purpurea is a parasite that gets into rye and rye grain, and it produces the deadly toxin ergo. And then Cryptococcus can cause respiratory infections and meningitis if it gets into the nervous system, into the central nervous system. So here are some pictures of, like here, just what we think of as the mushroom out there in the wild. And here we are looking at a gill from the underside of a mushroom and the basidiospores that are growing in clusters of 4 as a result of meiosis in that diploid cell that forms after the 2 nuclei fuse. And then last of all are the Deuteromycota, sometimes known as the Fungi imperfecti. And they're considered imperfect because they don't have a sexual stage, or at least we have not been able to discover a sexual stage in their life cycle. And so because of this, they can be difficult to determine where they go taxonomically, but most of them really resemble the Ascomycota. And so as we do more exploration looking at their DNA, then, over time I suspect that we will see the Fungi imperfecti become a smaller and smaller group as we're able to place these different organisms where they belong. Okay, so I think that's everything I got to tell you about the fungi. Let me double check that there's not